All right, we're starting the second part of the Chapter 4 notes on the structure and function of eukaryotic and prokaryotic cells. Again, focusing more on the prokaryotic cells. Our couple of objectives for this section of the notes is to be able to compare and contrast the structure and function of gram-positive and gram-negative cell walls. This is very important because that really determines how well different antibiotics work against it. And so this week we're going to be doing a lab where you'll practice doing gram staining, and then we'll finish the week with doing a lab on antibiotic resistance and how well different antibiotics work on gram-positive versus gram-negative bacteria. So when we look at the functions of the cell wall, remember that it's a semi-rigid structure, much more firm than a cell membrane is, and so it gives shape to the cell. It helps protect the inner plasma membrane or cell membrane. It can prevent the bacterial cell from rupturing when you have high water pressure. It helps anchor the flagella for those bacteria that have a flagella. And in some cases, it contributes to the virulence of some bacteria. So how disease-causing they are and how bad of a bacteria they are, basically. It's the site of action of some antibiotics. So as we talk about some of those antibiotics, they're going to try to attack the cell wall. And then what's important to us here is that the chemical composition can help differentiate types of bacteria. And so we always talk about gram-positive and gram-negative bacteria. That's one of the easiest two big categories we can divide bacteria into, and it's based on how they stain, how their cell walls are composed. And so we'll be really focusing on that in this section. So if you remember from the last chapter that the cell walls of bacteria are composed of a substance called peptidoglycan. And these are repeating disaccharides made up of, I'm going to call them NAG and NAM, N-acetylglucosamine and N-acetylmuramic acid. And we'll show you that structure here in just a minute. But it's linked together by polypeptides. And one thing about it is penicillin, here's one of our first antibiotics, can interfere with the linkages. Okay, so here you have these peptide cross bridges here, that's the blue and green here, holding the peptidoglycan there, the NAG and the NAM sections of it. What can happen is penicillin can interfere with those linkages and cause cell lysis. That means that the plasma membrane ruptures. Well, if it ruptures, it's going to lose all the liquid that's inside, all that cytoplasm, along with any kind of food and materials and nutrients and so on that we need in there, and specifically water, and that's going to cause death of that bacteria. So that's one of the ways that one of the antibiotics works, is it interferes with the cell wall linkages there, those polypeptide linkages. So gram-positive cell walls have a very, very thick peptidoglycan layer. Okay, they also have what are called teichoic acids made up of alcohol and phosphate, and these may regulate movement of positive ions in and out of the cell, regulate cell growth. They also allow for identification of gram-positive bacteria through certain lab tests as well. But here the big thing is this very thick peptidoglycan layer. Gram-negative cell walls have a very thin peptidoglycan layer, but they have an outer membrane also that's found on the outside of that peptidoglycan layer. It's composed of lipopolysaccharides, lipoproteins, and phospholipids. So here's the peptidoglycan layer right here, made up of that NAM and NAG. And here's your actual cell membrane or plasma membrane, but here's an actual outer membrane that the gram-negative bacteria have. And it looks almost just like your plasma membrane, because it's a phospholipid bilayer, just like the other layer is. Now that outer membrane provides protection from phagocytosis, complement, provides a barrier to antibiotics. So all of these help protect gram-negative bacteria really from attack of your immune system or any antibiotics you might take. And so they're a little bit tougher sometimes to knock down and attack because they have that extra outer layer. And then they can have porins, which are just basically channel proteins that allow movement of materials through the cell. Some of them have what's called lipid A, which is an endotoxin, so that can add to their virulence, how much disease-causing they are. And then they can also have the O polysaccharide antigen. Okay, remember the O157? That's one of the antigens. I remember the flagella had the H antigen in it, and that added, kind of signal that particular type of E. coli in this case, for example. So here, if you just look at the layers here, you see this brown layer here? That's the thick peptidoglycan layer. And see this little thin layer here? That's the thin peptidoglycan layer, plus they have this outer membrane here. So that's kind of the differences between the gram-positive and gram-negative cell walls. So why is this important? Well, we do what's called a gram stain, and we can actually, by doing this step-by-step staining process, we can actually see whether your bacterium is gram-positive or gram-negative based on how it stains. And so there's 4 basic steps. We have the first step, which is called crystal violet, which is the primary stain. That means it's the first stain. That dye is going to enter the cytoplasm, and it's going to stain both gram-positive and gram-negative cells at that point purple. And then we use what's called Gram's iodine, as opposed to just regular old iodine. And that acts as the mordant, or the kind of fixer. It kind of fixes it in place. What it does is it causes the large crystals to form in the dye, so it can't leave the cell wall. It'll have difficulty leaving the cell wall. So it's going to stain both gram-positive and gram-negative purple still. They're still going to have that crystal violet color because of those crystals forming in there. Then we add what's called ethyl alcohol, which is a decolorizer. What it does is it dehydrates the peptidoglycan layers of gram-positive cells, making it more difficult for that crystal, for those crystals to leave. But it dissolves the outer membrane of gram-negatives, okay? And once it dissolves that outer membrane, that crystal violet color leaves. So at this point, your gram-positive cells will look purple and your gram-negative cells will look clear. Well, if you try to look at a bacteria that's clear in the microscope, are you going to see it? No. You can hardly see it when it is stained, but if it's not stained, you're not going to see anything. So we have to use what's called a counterstain. In this case, it's safranin, and it provides a contrasting stain to the primary stain. And so this is more of a reddish pinkish colored stain. So then a gram-negative in the end, because remember it was clear up here, now it's going to look pink. Basically, it'll take on the color of that safranin, that counterstain. Well, pink doesn't really cover up a dark purple, so the gram-positives will still look purple because of the crystal violet that's really stuck in that thick peptidoglycan layer. And so, really, these are the 2 of what you're going to look for once you get done to see is your bacterium gram-positive, gram-negative. Now, maybe you already, when you did your research for your wanted poster, found out whether it's gram-positive, gram-negative. Well, you'll be able to do the gram stain to see if you can confirm your results. So here we have application of the crystal violet dye. Both types will look purple. Then we add the iodine, the mordant. Again, both will look purple. Then we use the alcohol to kind of decolorize it, and the gram-positives will still stay purple, but the gram-negatives will look clear. And then we'll use the counterstain, the safranin, and now the gram-negatives will look pink, and the gram-positives will look purple. So you can see this is what it will kind of look like, and you'll be able to see shapes, like you can see that kind of almost like a spirilla here, or vibrio almost, and then you can see some gram-positive rods there. Some of them are diplobacillus because they're in little things of 2, some of them are in long chains, so maybe they're streptobacillus. But here, gram-positive will look dark purple, gram-negative will look a light pink. Some organisms have atypical cell walls, like acid-fast cell walls. They're kind of like gram-positive cell walls, but they have a waxy lipid area made up of what's called mycolic acid that's kind of connected to the peptidoglycan layer. These are just a couple of genuses, Mycobacterium and Nocardia. We can't order those into the school, so we don't really cover these. Some of the mycoplasmas don't even have a cell wall, but they have sterols in their plasma membrane that kind of help protect them from lysis. And then, of course, the archaea, that includes all the ancient bacteria, not all the bacteria that we work with. These do not have cell walls, or if they do, they're made of a substance called pseudomurine. So they won't have this, the NAM or the other amino acids. These cannot be gram stained because they'll just look gram negative because they don't have that peptidoglycan layer. So it doesn't do any good to use a gram stain on them. We only use those on just the regular bacteria that have peptidoglycan layers. As far as damage to the cell wall, lysozyme, which can be found in your tears, can digest the disaccharide in the peptidoglycan layer. Again, we already mentioned that penicillin inhibits the peptide bridges in the peptidoglycan layer. A protoplast is a wall-less cell. It doesn't have its wall around it. A spheroplast is a wall-less gram-positive cell, and these are more susceptible to what we call osmotic lysis. That just means if too much water comes in and builds up, it may cause that cell to rupture if they do not have a cell wall. And other structures called L-forms are wall-less cells that swell into irregular shapes, so they won't look like the more common coccus or bacillus like the other cells will. So that kind of takes you through the cell wall.